What if we could actually pinpoint the exact genes that control how we age? Well, believe it or not, a group of researchers might have just pulled it off. They've identified 6 specific genes that seem to be the master regulators of aging, not just in one species, but across several. This is, and I'm not exaggerating, a huge leap forward in our quest to understand one of biology's greatest puzzles.
But to really get why this is such a big deal, you have to appreciate just how messy and complicated studying aging is. I mean, it's not a single neat process. It's chaos. Different parts of the body age differently, and the genetic data, it's just a sea of noise and random signals. Trying to find the real master switches and all of that is like trying to hear a whisper in a hurricane.
So the scientists behind this discovery had to get clever. They took an approach that's kind of like tuning an old radio. They were convinced that somewhere in all that static, a clear signal for aging existed. The trick wasn't just to listen harder, but to find a brand new way to filter out all that background noise and lock on to that one consistent repeating pattern.
So what was their secret? What was this clever shortcut? Well, instead of getting stuck in one single massive experiment, they decided to zoom out. They looked for genes whose activity levels consistently change with age across a whole bunch of different species and tissues. The idea is simple, but it's really powerful. If a gene's activity does the same thing in mice and in rats and in humans as they get older, well, that's a pretty darn good sign it's a major player in aging.
And when I say they zoomed out, I mean it. They didn't just look at 1 or 2 studies. They pulled the data from 25 different studies on gene expression in mammals. Think about that. They created this enormous data set to hunt for those repeating signals. This was the very top of their discovery funnel, casting the widest net possible.
So how do you sort through all that data? They use something called a value counting method. You can just think of it like a popularity contest for genes. They scored each gene based on how many times its activity went up or down with age across all those 25 studies. The ones that kept showing up, behaving the same way time and time again, they rose right to the top. And this filter was incredibly powerful. It took thousands of possibilities and narrowed them down to just 19 top suspects.
Okay, so now they have a list of 19 potential culprits. But that's just a list of suspects, right? You still have to prove they actually did it. The next and absolutely crucial step was to take these 19 genes and test them in a living creature to see if they genuinely, for real, affect how long it lives.
And for this, they turned to a true hero of aging research, the tiny roundworm, C. elegans. Now, why these little guys? Well, their lifespan is super short, so you can see the results of an experiment in just a few weeks instead of waiting for years. The researchers used a technique to turn down the activity of each of those 19 suspect teens. And here's the really smart part, they only did it after the worms were already adults, so they knew they were looking at aging, not weird developmental effects.
And the results? They were absolutely stunning. Of the 19 genes they tested, dialing down 6 of them consistently made the worms live longer. 6 out of 19. That's a success rate of over 30%. In this kind of genetic screen, that is almost unheard of. They hadn't just found suspects, they had confirmed hits.
So let's meet the lineup. Who are these 6 genes? Well, you've got some familiar faces here. CASP1, that's a well-known bad guy involved in inflammation. You know, that chronic low-grade irritation that causes so many problems as we get older. SPARC is also tied to inflammation, but then you get some surprises. DIRC2 deals with the cell's waste disposal system. RSRC1 is involved in RNA splicing, which is like the editor for our genetic code. It's this fascinating mix of the usual suspects and some brand new persons of interest.
But wait, because buried in these results was something truly weird. A twist that kind of challenges how we think about aging at a genetic level. So get ready, because biology just threw a major curveball.
Let me just ask you this. If I told you a gene's activity naturally goes down as you get older, what would your gut instinct be? You'd probably think, oh, that must be bad. We should try to boost it back up, right? That's the logical assumption. But what if the complete opposite was true?
Well, take a look at this. It's just wild. Of the 6 genes that made worms live longer when turned down, only 2 were genes that normally go up with age. The other 4, they were genes that were already declining with age. So turning them down even more was the key. It makes no sense at first glance. It's totally counterintuitive.
And that leads to this massive takeaway. We just can't assume that genes that go up with age are bad and genes that go down are good. It's not that simple. The body's response to getting older is way more complex. Maybe some of these changes are just the body trying and failing to fix things. This discovery really forces us to rethink what these genetic changes actually mean.
So, of course, the billion-dollar question is, what does all this mean for us, for people? I mean, a result in a tiny worm is a long, long way from a pill you can take. But what this research does is give us some incredibly strong clues about which of these 6 genes are the most promising ones to go after in mammals, like us.
So, SPARC and CASP1, they're already the frontrunners. We've got pretty strong evidence in mice that turning them down helps fight that age-related inflammation. CA4 is also a really hot prospect, mostly because it belongs to a family of proteins that we already know how to design drugs for. As for the other 3, they're a bit more mysterious, but this new discovery has just shoved them right into the scientific spotlight.
Now, it's really, really important we're clear about what this discovery is and what it isn't. This is not the fountain of youth in a test tube. It's not a ready-to-use therapy. What it is a powerful tool, like a super-powered searchlight that gives scientists a much shorter, much higher quality list of places to look. It's a way to dramatically speed up the entire process of discovery.
And that, right there, is the real heart of it. The biggest value of this study isn't that it found a magic bullet for aging. It's that it gave us a much, much better map of the territory. It tells researchers where to focus their time, money, and energy for the best possible chance of finding something that works.
And if you want to see just how big a leap forward this is, check this out. A typical old-school genetic screen might get a hit rate of maybe 1 or 2% if they're lucky. This new cross-species method, it had a hit rate of over 30%. That is a staggering improvement in efficiency. We're talking about saving years of work and millions of dollars in research.
I think this quote just nails it perfectly. This research doesn't hand us the treasure. It hands us a really sharp, accurate map that says X marks the spot. Dig here. And in a field as vast and confusing as aging, having that map might just be the most valuable discovery of all.
So it leaves us with this really exciting question to think about. This study didn't just find 6 genes. It proved a whole new way of finding them is possible. Now that we have this incredible method for tuning into the core signals of aging, what other fundamental secrets are we about to uncover next?